Welcome dear students. In this video we are going to learn about Glycerol-3 phosphate shuttle. In my previous video I have already discussed about malate aspartate shuttle, and in that video I have mentioned that this shuttle, malate aspartate shuttle, actually occurs in the cardiomyocytes and liver. Now this Glycerol-3 phosphate shuttle actually occurs in the skeletal muscles. So at first I want to mention another time that what is the reason of this shuttle. So the main function of this shuttle is for the glycolysis, because the glycolysis process occurs in the cytoplasm, and in that glycolysis 2 ATP molecules and 2 NADH molecules are produced. Now this NADH molecule is the reason for this shuttle, for the malate aspartate shuttle and for the Glycerol-3 phosphate shuttle. And the reason is because this NADH is actually high energy electron carrier. One NADH molecule can carry 2 high energy electron that actually take part in the electron transport chain, and indirectly it helps in the generation of ATP molecules. Now the problem is here, there is a two problem. The first is, in the cytoplasm there is a NAD plus that is converted into NADH and H plus. Now the problem is here that to continue the glycolysis mechanism, continuous supply of NAD plus is required. Continuous supply, because this NAD plus actually gets reduced to NADH. Eventually it actually carries the high energy electron. So to continue this glycolysis, continuous supply of NAD plus is required. But as we all know that the cytoplasm have a limited amount of NAD plus, so this NAD plus should be regenerated by oxidation of NADH. So this is the first reason for this shuttle. Another reason is these two molecules, NAD plus and NADH, is actually impermeable into the matrix, mitochondrial matrix, because the inner mitochondrial membrane is actually impermeable for these two molecules. So thus in the cytoplasm, those NADH molecules are generated cannot enter directly into the mitochondrial matrix. And we all know that this TCA cycle and the electron transport chain actually occurs in the mitochondrial matrix or inner membrane of the mitochondria. So there is no direct access of this NADH into the mitochondrial matrix, and there is necessary of regeneration of NAD plus to continue the glycolysis. So in that situations, cell plays a trick. In this case, this is called Glycerol-3 phosphate. Here a molecule called DHAP, full form is dihydroxy acetone phosphate. This molecule is actually a intermediate metabolites in the glycolysis mechanism, glycolysis pathway. I have already mentioned that the fructose 1,6-bisphosphate get actually cleaved into two products, two triads we call DHAP, dihydroxy acetone phosphate, and Glycerol-3 phosphate. In this case, the DHAP is actually a intermediate metabolites in glycolysis. Now the DHAP can be reversibly converted into a product called Glycerol-3 phosphate. That's why this shuttle is called Glycerol-3 phosphate shuttle. Glycerol-3 phosphate. So the DHAP is reversibly convertible into Glycerol-3 phosphate, and the enzyme here is Glycerol-3 phosphate dehydrogenase. This is the enzyme, dehydrogenase. Now you can understand, in my previous many videos I have started to mention that those enzymes named as a dehydrogenase, that means it can exchange hydrogen atom with the molecules. Now here the hydrogen molecules actually come from NADH plus H plus, that is actually generated in by this glycolysis I have mentioned already. So this NADH that is actually generated in the cytoplasm. So this is actually cytoplasm. So the cytoplasm, in the cytoplasm those NADH are actually generated cannot enter inside the mitochondrial matrix directly. So this NADH gets actually oxidized to NAD plus. So first criteria fulfilled, that the NAD plus gets replenished or regenerated, so then the glycolysis can continue. So this is the first reason. And another important reason is this NADH carry 2 high energy electrons that is actually passed through the DHAP to glycerol-3 phosphate. Now the glycerol-3 phosphate have two high energy electrons that it carries. Now the glycerol-3 phosphate can enter inside the mitochondria. So in the mitochondria there is two membrane. Let's suppose this is the first mitochondrial membrane, the outer mitochondrial membrane. So I am writing here on the outer membrane, and there is a inner membrane. This is the inner mitochondrial membrane. Okay, so the glycerol-3 phosphate can enter, this molecule can enter inside the mitochondria. But now you have to understand this molecule do not pass through the inner membrane. It actually remain here. It is called inner membrane space, intermembrane space. So in between this outer membrane and inner membrane, this is the matrix. So in between this outer membrane and inner membrane, there is the intermembrane space. This molecule, this glycerol-3 phosphate can enter and remain here, glycerol-3 phosphate. So now this glycerol-3 phosphate then reversibly converted into DHAP. Okay, get reversibly converted into DHAP, dihydroxyacetone phosphate, and this DHAP actually come out from the intermembrane space. So the enzyme that actually take place in this reaction is actually the glycerol-3 phosphate dehydrogenase, but it is membrane bounded in conditions. You have to understand this. This is the same glycerol-3 phosphate dehydrogenase. So this enzyme is the cytoplasmic version of this enzyme, and this is the mitochondrial version of glycerol-3 phosphate dehydrogenase that is actually embedded in the inner mitochondrial membrane. So this enzyme actually converts. Now what happens in this glycerol-3 phosphate dehydrogenase, there is a molecule called FAD. This is also an electron carrier. Now the same thing occurs, as I have mentioned, that the glycerol-3 phosphate now contains the two electron, high energy electron. It actually donates this electron and the proton, and now the FADH2 actually becomes FADH2. Okay, you have to understand this. When the glycerol-3 phosphate reversibly converted into DHAP, it releases the electron and the hydrogen atom, and it makes the FADH2. FADH2 is actually already present in this enzyme as a cofactor. It receives the high energy electron and converted into FADH2. Now what happens here? A ubiquinone is present. This is also another electron carrier. So Q means ubiquinone. This ubiquinone can receive high energy electron and proton. Then this ubiquinone is converted into dihydroquinone. Always it is also known as ubiquinol. Okay, so it becomes Q to QH2. That means it has actually become reduced, and it carry the proton. Now this QH2 actually perform the main function in electron transport chain. So if you want to understand this subtle mechanism in case of malate or in case of glycerol-3 phosphate subtle mechanism, you have to understand what is electron transport chain occurs. So I will take a separate lecture on this electron transport chain, but to make this mechanism understandable, I will show you a general mechanism of this electron transport chain. What happens here, I will show you just briefly. So let's suppose this is inner mitochondrial membrane, because we know that this all electron transport chains are actually embedded molecules. This is an integral membrane protein that is present in the inner membrane of the mitochondria. So in this electron transport chain, there are 4 complexes are actually present. Let's suppose this is the complex 1. This is the complex 1. Then another complex is present called complex 2. There is another complex, complex 3. Another complex, complex 4. And ultimate another enzyme is actually present. We call it, let's suppose this is an enzyme. one enzyme is, we call this enzyme ATP synthase. ATP synthase. Okay, so basically what happens, there are two routes of these electron transportations. We call it complex 1 to complex 3 to complex 4 and ultimately to the ATP synthase. There is another route the electron can transfer from one complex to another. Is called complex 2 to complex 3 to complex 4, so and ultimately to the ATP synthase. So when electron pass through this let's suppose here pass through this complex 1, then it actually bypass the complex 2 and it reach to the complex 3 and then by actually go to the complex 4 and actually ultimately it actually released into the water molecules, oxygen molecules to reduce into water molecules. So I'm not going into details, but I'm just making you clear that the role of this glycerol-3 phosphate and ubiquinone. So what happens, I have mentioned in my malate aspartate shuttle mechanism that in that shuttle actually the one NADH molecule that is present in the cytoplasm actually converted into the matrix a one NADH molecules plus H plus molecules. If you have not watched my video, must watch the video, then you can understand that one NADH molecules is actually converted into the inside into the matrix in NADH and H plus. When NADH actually take part in the electron transport chain, it starts its journey, the electron actually starts its journey from complex 1 to complex 3, then complex 4. You have to understand this first. When NADH releases electrons, it starts from the complex 1, then complex 3, this complex 4. When electron actually pass through complex 1, 4 hydrogen atom, 4 H plus atom protons actually released from the complex 1. When electron pass through the complex 3, 4 H plus atoms are released from the complex 3, 2 directly and 2 hydrogen from the quinol. Okay, quinol. So another, when the electron pass through complex 4, it actually helps in the transportation of 4 hydrogen protons. So now you can see when the starting materials is NADH, that is in the case of malate aspartate shuttle, then 12 protons are actually transported out of the matrix to the intermembrane space, and that 12 hydrogen actually participate in the generation of proton motive force. Now when glycerol-3 phosphate, so this is the result of malate aspartate shuttle. Okay, for one NADH 12 proton will be actually released. Now when the glycerol-3 phosphate shuttle actually occurs, what happens here? Ubiquinol or dihydroquinone is already produced. Now I have to give you one actually information that when electrons are actually transported from NADH plus H plus from the complex 1, actually what happens, a quinone, ubiquinone actually ultimately carry the high energy electron to the complex 3. This is actually the process what happens here. In case of electron transport chains, when NADH actually release the high energy electron, it actually pass through the complex 1 and ultimately is carried, actually taken by the ubiquinone, and this ubiquinone actually can transfer from the complex 1 to complex 3. So this is the case of NADH, but here you can see in the glycerol-3 phosphate shuttle already a ubiquinone is produced. So when glycerol-3 phosphate shuttle is actually taking place, then this complex 1 is actually bypassed. You have to understand this. The complex 1 is actually generate QH2. Now this enzyme is actually generating the QH2, so there is no need of this complex 1 because in the glycerol-3 phosphate shuttle there is no generation of NADH in the matrix, so there is no NADH, so the complex 1 will be actually bypassed. So actually what happens in case of glycerol-3 phosphate, as the complex 1 is bypassed, so there will be no hydrogen atom will be released from this complex 1, only 4 and 4 8 protons will be released by this glycerol-3 phosphate shuttle. And here is the difference why the malate aspartate shuttle is more efficient in generation of proton motive force and ultimately in the generation of ATP, because in this case less amount of protons are actually released. So if you want to understand this mechanism in more details, you have to know the facts about the electron transport, and I have just given a very basic idea, because we know that this proton motive force that is actually generated by the pumping of this proton from the matrix to the intermembrane space, actually that force actually helps in the generation of ATP by the synthetase enzyme. So by this way, if there is low amount of protons are generated, protons actually pumped out, then low amount of ATP will be generated. That is why glycerol-3 phosphate is inefficient than the malate aspartate shuttle. But by the way, this actually can fulfill the two needs of the cell to regenerate the NAD plus and to carry out the high energy electron to the mitochondrial matrix. So this is all about glycerol-3 phosphate shuttle. Hope this video will help you. Thank you for watching, and always keep learning.